Hi all. Today we will be discussing on architecture of life. Every living organism is made up of cells, right? Which are the fundamental units of life. Cells perform all the essential processes required for the survival, growth and reproduction. So although cells vary in size, function, etc., they are share many common structures that work together to maintain life. So here we will explore the major organelles found in eukaryotic and prokaryotic cells and understand how each contribute to the functioning of the cell. Okay, so let's begin. Start with the structure of cell surface. Okay, so the cell surface membrane is also known as the plasma membrane. It is an extremely thin structure, approximately 7 nanometers thick. Okay, it consists of a phospholipid bilayer with the hydrophilic phosphate heads facing the aqueous environment inside and outside the cell. So what do it have? It has a phospholipid bilayer. Okay, lipid bilayer. Okay, both in inside and outside of the cell. And the hydrophobic fatty acid tails facing inward. So these are the heads and these are the tails. Okay. The membrane serves as selective barrier. So what is its function? It acts as a strict barrier and controls the movement of substances inside and out, right? So controlling the movement of substances into and out of the cell. And it also plays an important role in signaling, cell signaling. Okay, that is the communication, right? How does it do that? Through receptor proteins, cell recognition through surface antigens and cell adhesion. So it enables signaling via receptor proteins and cell recognition uh, via cell surface antigens. Okay. Additionally, enzymes that have a lot of addition activities, that is physical interaction, facilitates cell to cell addition and angers the cytoskeleton. Okay. And what is the catalysis here? What is the catalysis? The enzymes embedded within the membrane. They catalyze reactions while the membrane anchors the cytoskeleton and maintains cell stability. Okay, these are the basic things about cell surface membrane. Now let's move forward to the nucleus and nucleolus. So the nucleus is often called the control center of the cell, right? Why? Because it contains the genetic material that directs all the cellular activities. It is surrounded by a double membrane known as the nuclear envelope, right? So it is called as nuclear envelope. It has a double membrane, right? It is embedded within the envelope are the nucleopores that regulates the exchange of material. So basically it embedded within the envelope, right? So you can see here. And there is a lot of nuclear pores visible here, right? So there are messenger RNA and ribosomal subunit leave the nucleus while proteins, ATP, nucleotides and regulatory molecules enter. So what are the things leaving to the nucleopores? The messenger RNA, ribosomal subunit. And what all things enter? Proteins, ATP, that is energy, nucleotides and hormones. Okay. Inside the nucleus are chromosomes. Okay, inside the nucleus what we have? Chromosomes. What are chromosomes? They will control the cell activities and inheritance, right? So which contain DNA organized into genes. So these genes carry instructions for protein production and inheritance. And then we have the nucleolus, right? There is a dense region within the nucleus that manufactures ribosomes, which are essential for protein synthesis. So it contains DNA or RNA that produces the ribosomes. Clear? Now let's learn about the ribosomes, the translators. So ribosomes are the sites of protein synthesis, right? So their main function is to translate the genetic instruction carried by messenger RNA into chains of amino acids called polypeptides. Okay. So what they do it is a site of protein synthesis. They translate mRNA into polypeptide using rRNA and proteins. Right. So each ribosome consists of a large and small subunit made from the ribosomal RNA and proteins. The small subunit bind to the messenger RNA. Okay. While the larger unit joins amino acid together. So the larger unit will join the amino acid together. Okay. So in eukaryotic cell, there are 80 small ribosomes. Okay. Located in the cytoplasm and attached to the rough ER, that is standard eukaryotic cell. Okay. So it is attached to the rough endoplasmic reticulum. Smaller 70S ribosomes are found in the mitochondria. Okay. 70S ribosomes are found in the mitochondria and chloroplast in the smaller variant. Okay. And resemble those found in the bacteria. So these are ribosomes. Okay. Now let's see the synthesis, manufacturing and shipping hub. That is the rough ER and Golgi apparatus. Right. So here let's see the pathway followed by proteins after synthesis. What will happen to the proteins once it is synthesized? So protein production begins on ribosome as we have learned before, right? It attached to the rough endoplasmic reticulum or RER. Okay, so these newly synthesized proteins enter in the sacs of the RER where they are folded and modified. So they are formed here, right? So the rough ER and smooth ER. We have two. So the network of membrane sac covered in ATS ribosomes, that is RER, synthesized and transport newly built proteins. So the tubular membrane without ribosome, that is SER. Okay. So they synthesize lipids and steroids like cholesterol, sex hormones, etc. Okay. And then it has to be transported, right? So the small membrane bounded sphere that ferries proteins and lipids from ER to the Golgi apparatus. That is the transport vesicle. Okay. So transport vesicles then carry this protein to the Golgi apparatus. The Golgi body act as like the processing and packaging center, modifying proteins. They will sort them and also preparing them for the final destination. So the Golgi apparatus. It is a stack of flattened cystine, right? So it receives vesicles from RER and SCR. Okay. So it modifies proteins or lipids and it will either sort it or package it, the molecule, and produces Golgi protein and lysosomes. Finally, what will happen? The vesicle bud off from the Golgi apparatus and transport the protein either to other parts of the cells or outside the cell through exocytosis. Okay, the last process is exocytosis. So secretory vesicle produce uh, the Golgi fuse, right? So the secretory vesicle that produced by the Golgi fuse with the plasma membrane and releasing contents inside the cell. Okay, so this is what happens. First it is produced, then it is transported, and then it is moved through the exocytosis, right? Lysosomes. They are also known as the recycling center. Okay. So lysosomes are membrane-bound organelles that contain powerful hydrolytic enzymes. Okay. So these enzymes are capable of breaking down proteins, lipids, carbohydrates, and nucleic acids. Okay. So the primary role is intracellular digestion. So what is their key function? Intracellular digestion. So lysosomes remove damaged organelle. Okay, they will digest the worn out organelle and will break down the uh, cellular waste and destroy the harmful microorganisms engulfed by white blood cells. Okay, so they will break down the waste material, destroy the invading pathogens. Okay. So because these enzymes are potentially destructive, they are enclosed within a membrane that prevents them from damaging the rest of the cell. So it is very important, right? Since it is destructive in nature, if it is get out of hand, it will be bad for the body. right? So we have to be very careful here. That is why it has the protection layer. Okay, the protection membrane. Clear? So basically it is a single membrane containing powerful hydrolytic enzymes. Okay, and it is formed by the Golgi. So this is the lysosome, that is a digestive vesicle, right? So they engulf the cellular debris. Okay, and this with the help of the enzymes, it will digest them. Clear? Then we have the mitochondria and ATP generation, right? So mitochondria is called as the powerhouse of the cell. Let's see why. So mitochondria are often referred to as the powerhouse of the cell because they produce most of the cell's ATP through aerobic respiration. So we have two type of respiration, right? Aerobic and anaerobic respiration. So in aerobic respiration, the ATP is produced through mitochondria. So each mitochondrion is surrounded by a double membrane. This inner membrane is folded into structures called cristae. Okay, so we have this structure called the cristae, which greatly increase the surface area available for energy production. So the matrix contains enzymes, cellular the circular DNA and the 70S ribosomes, etc. ATP or adenosine triphosphate serves as the cell's energy currency. Okay. So energy is stored in the phosphate bonds and released when ATP is broken down into ADP and inorganic phosphate. So this energy powers the numerous cellular processes. So these are the mitochondrions. Okay. We have a number of mitochondrion. And what is the outer membrane? It contains protein, right, the porine proteins. Then inner membrane, highly selective, folded into cristae to exponentially increase the surface area for aerobic respiration. Okay. Then it have internal intermembrane space, the gap between the membranes, right? And there is a matrix that contains respiratory enzymes, circular DNA, and 70S ribosomes synthesizes the lipids. Okay, so it act as the energy currency in the cell. Okay, for the function of the cell. Clear? Now let's check the chloroplast. Okay, so chloroplasts are specialized organelle found in plant cells. So we saw mitochondria in animal cell. Now we have the chloroplast in plant cell and are responsible for photosynthesis. So we have learned this before, right? In photosynthesis, we need chloroplast. Within chloroplast, there are flattened membrane sacs called the thylakoids. So we have the thylakoids here, right? The flattened membrane. These thylakoids contain chlorophyll, the pigment that capture light energy. So thylakoids are sacked into structures called grana. So you can see the grana here, right? And which are connected by lamellae. So the connection is called the lamellae. You can see that here. So the fluid surrounding this structure is called as the stroma. Okay, we have the stroma and it contains enzyme, circular DNA and 70S ribosomes. So during photosynthesis, chloroplasts convert light energy into chemical energy and stored in the form of glucose. Okay, so this is what the function happening in chloroplast. Clear? Now let's see the endosymbiotic theory. Okay, so the endosymbiotic theory explains the evolutionary origin of mitochondria and chloroplasts, how they originated. Okay, so how did they originate? Any idea? So, according to this theory, these organelles were once free living bacteria that were engulfed by a larger ancestral cells. So, instead of being digested, they formed a mutually beneficial relationship. Okay. So, several pieces of evidence support this theory. So, both mitochondria and chloroplast possesses double membrane, circular DNA, 70S ribosomes, and the ability to replicate independently of the cell. So, these characteristics closely resemble to those of modern bacteria. Okay. So, the double membrane structure, their genetics, machinery, and reproduction capacity. Clear? Now, let's see the microtubules, the structural scaffold. So, microtubules are long, hollow cylindrical structures that form part of the cell's cytoskeleton. They are composed of alpha and beta tubulin protein that join to form dimers. Okay, clear? Alpha tubulin and the beta tubulin that forms the dimer. So these dimers are assembled to protofilaments. Okay, they assemble to protofilaments. See the protofilament. And 13 protofilament combine to form a microtubule. So 13 protofilaments together forms a microtubule. So microtubules help maintaining cell shapes, support the intracellular transport, guide the movement of organelles and vesicles, and form spindle fibers during cell divisions. Okay, so they also provide structural support for cilia and flagella. So these are the important points, the functions. Okay, so maintaining the cell shape, providing tracks for intracellular transport, moving organelles or vesicles, form spindle fibers for cell division, and enabling movement of cilia and flagella. Okay, clear. Now let's see the chromosomes and cilia. So we will compare the structures of centrioles and cilia here. Okay. So centrioles are cylindrical structures composed of 9 triplets of microtubule. Okay. So they have 9 triplets of microtubule, as you can see here. Okay. So made up of 9 triplets. Clear. They are found within the centrosome and organize spindle fibers during the cell division. So two centrioles sit at the right angles inside the centrosome, and the main microtubule organizing the center. Okay. Their function: organize the spindle fibers, help chromosome separation during cell division, and forms the basal bodies. Okay. And what about cilia? They possesses a distinctive 9 plus 2 arrangement. As you can see here, it is a 9 plus 2 arrangement consisting of 9 outer microtubule doublets. Here it was triplets, here it is doublets. See? Okay. So this arrangement enables coordinated movement. So in the respiratory tract, cilia help move the mucus and trapped particles away from the lungs, protecting the body from infection. So the structure is hair-like projections using the 9 plus 2 microtubule, right? The double arrangement. And what is the function? Move substances across cell surfaces and for cleaning the respiratory tract, moving the eggs in oviducts, etc. Clear? Now let's see another form, that is villi, the microvilli. Okay, so microvilli are tiny finger-like projections extending from the cell surface. Unlike cilia, microvilli do not move. Their primary function is to increase the surface area, right? Allowing more efficient absorption and secretion. So they are especially abundant in the small intestine, where they increase nutrient absorption, and in kidney tubules, where they enhance reabsorption of water and solutes. Okay. So what is their function? Dramatically increase the cellular surface area and to improve absorption and reabsorption efficiency. They are mostly found in small intestine and kidney tubules. Then we have the cell wall and vacuole. Okay. So the plant cell possesses unique structure not found in animal cell. Okay. The cell wall is composed primarily of cellulose and it provides rigidity, protection and support to the cell. Okay. It prevents the cell from bursting when water enters through osmosis and help maintain the turgidity. So the large central vacuole, you can see here the large central vacuole occupies much of the plant cell's volume. It contains cell sap, which stores sugars, pigments, mineral ions, waste products, and water. So the surrounding membrane, known as the tonoplast, that regulates movement into and out of the vacuole and help to maintain the osmotic balance. Okay, so what is the cell wall? It is rigid, freely permeable outer layer composed of mainly cellulose, right? And then it provides the shape, prevents bursting, and maintains the turgidity and offers physical protection. And we have Plasmodesmata, that is specialized channels connecting adjacent plant cell for communication and transport. And what about the vacuole and tonoplast? It is a vacuole is a large central storage sac surrounded by a specialized membrane called a tonoplast. And the cell sap inside it stores the water, sugars, enzyme, mineral salt, pigments, and waste. What is the function? Maintains turgor pressure and regulate the osmotic balance. So it is also very important, right? Now let's check the two type of cell, that is prokaryotes and eukaryotes. So here we will compare the two major categories of the cells. So prokaryotic cells such as bacteria are relatively simple. They lack a true nucleus and membrane bound organelle, right? So their DNA is circular and lies freely within the cytoplasm. But what about eukaryotic cells? They are found in animals, plants, fungi and protists. And these are more complex. Okay, so they possesses a true nucleus containing linear chromosomes and variety of membrane bound organelle that compartmentalize cellular functions. So these structural differences contribute the greater complexity and specialization observed in eukaryotic organisms. Clear? Now let's see the metrics. So here we will see the major differences between prokaryotic and eukaryotic cells. Okay. So as you can see, the cell size, it is around 0.5 to 5 micrometers, right? Whereas eukaryotic range from 10 to 100 micrometer. And prokaryotics contain circular naked DNA and 70s ribosome. Okay. And what about eukaryote? They contain linear DNA associated with the histones and the ATS ribosome. Okay. Then for the nucleus, it is absent. In eukaryotic, it is present. As we mentioned, ribosome is 70S, here it is 80S. And what about endoplasmic reticulum? In prokaryotic, it is absent. In eukaryotic, it is present. And organelle, in prokaryotic, there are only few or no membrane-bound organelles. But in eukaryotic, we can see many membrane-bound organelles. And about the cell wall, it is murine, right? Here it is cellulose and chitin. And what about cell division? They have the prokaryote have binary, but eukaryote they have mitosis and cytokinesis. And what about the transcription and translation? In prokaryote it is simultaneous, but in eukaryote it is completely a separate process, right? So what we have learned so far, cells are the fundamental building blocks of life, right? Each organelle performs specialized functions that contribute the survival and efficiency of the cell. Together, these structures create highly organized systems capable of growth, reproduction, communication, and adaptation. So, understanding cellular architecture provides the foundation for all advanced studies in biology and the medicine. Thanks for listening.